All right. Good afternoon, everyone. Thank you very much for coming to the colloquium this afternoon. I have the pleasure of introducing our speaker today, Dr. Alba Alfonso Garcia, who is a project scientist at UC Davis. Dr. Alfonso Garcia does a lot of different interesting biomedical imaging projects, one of which I have the pleasure of collaborating with her on. Today, she's specifically going to be telling us a little bit about fluorescence lifetime imaging and its connection to gastrointestinal health. And so, Alba, please take it away. Thank you again.
Yeah, thank you, Travis, and thank you everyone for coming this afternoon. So let's get started with this. The gut. The gut is so necessary, right? We need it for so many things. And sometimes it hurts. Sometimes it's just uncomfortable and it's nothing, but sometimes it can get serious. And so we need better ways to know what is happening there. Most of the diseases that we have in the gut are difficult to diagnose. A lot of things affect gut health. We have our lifestyles and habits can affect it. Environmental conditions, as well as pathogens, and obviously a multitude of other things. But what happens to this, to the gut when it's not good? Dysbiosis has been found as a central event in a lot of gut diseases, all the way from colorectal cancer to inflammatory bowel disease, but also associated with aging and inflammation. I should not be sorry. And as well, it's been also found in people who have a high-fat diet. What is dysbiosis? A colon is a normally hypoxic environment. There should be no oxygen there, and therefore it's populated by a lot of microorganisms that do not respirate oxygen. The moment microorganisms that can respirate oxygen and produce oxygen start appearing in there for one reason or another, they cause all sorts of problems. And typically they start blooming uncontrollably, and they cause inflammation in the tissue. So under these conditions, when you have bacteria and microorganisms that can both respirate oxygen and that cannot respirate oxygen, we decided that this is basically a dysbiotic colon, right? So what happens under dysbiosis? Under dysbiosis, the cells, the colonocytes, which are the cells that line the colon gut, the colon wall, change their metabolic profile. So they go from oxidizing fatty acid. So typically you eat your food, you get all your fibers, and you have all these microbes and bacteria that are breaking down this food, this fiber, into useful fatty acids for the cells to get their energy. And they just go into this oxidative fatty acids oxidative business, and it's a way for them to get energy, right? Suddenly, when you add an inflammation cue and you have a dysbiotic colon, this healthy microbiota disappears. You get these bacteria that can respirate oxygen all of a sudden, and you don't have those that can digest this fiber. So these fatty acids are all of a sudden not available. Your cells need to find another way to get energy, and they turn into a glycolytic state. They start burning glucose to create energy. And this is just one change, one of the possible changes that happens under dysbiosis. But that's an opportunity for us optics people, because with these changes a lot of things in their optical properties. The intrinsic optical properties of this tissue, of these cells, and therefore of these tissues change. As I said, the fatty acid content is going to be different. So you could think about maybe Raman spectroscopy. It's going to give us different signals. Another thing that changes is their fluorescence. The natural fluorescence of this cell, when it's undergoing oxidative phosphorylation, it's going to be different than those that are undergoing glycolysis. And this is a well-studied phenomena. It's been observed in microscopy studies, in cell culture studies, and organelles even with several groups. One of the things that changes with fluorescence is not only the color, which changes, but only slightly, but also the fluorescence lifetime. And this is what I want to talk about today. The fluorescence lifetime is going to be different for these two cells. And I'm just going to give you a very specific example right now, but bear in mind that this is not the only thing that happens when this metabolic change is undergoing. But there's a metabolic factor called NADH in our bodies that is part of many, many pathways. And we find this metabolic cofactor in different states. We can find it freely diffusing in the cytoplasm, and we can find it bound to a protein. When it's binding to a protein, there's a conformational change, and the fluorescence lifetime, which I'll explain in a minute what actually that means, is very long, okay? And so it's very characteristic of an oxidative phosphorylation state. Whenever the NADH is mainly freely diffusing in the cytoplasm, which is what happens in a glycolytic cell, the fluorescence lifetime quenches and it's very short. And so that could be an indicator that requires no labels. It's a label-free way, non-destructive way to see what's happening with your cells. And so I saw that back in the day. It's a little blurry still, right? But you can see that fine. Okay. I saw that back in the day with macrophages. This is a completely different system, if you will. This is not colonocytes. This is macrophages that I was studying in the context of inflammation. When you polarize your macrophages from a non-treated state, what they are called M0 macrophages, if anybody's familiar with them here, into a pro-inflammatory or anti-inflammatory phenotype, you actually are tweaking that same metabolic change that I mentioned in the colonocytes. And what we saw here, this is a way of representing the fluorescence lifetime, is that the lifetime of NADH when it's bound to proteins, in this case, it was bound to lactate dehydrogenase, is very long, about 3.4 in that case. When it's freely diffusing, it's 0.4 nanoseconds. And these two phasors here, so here, it's a representation of the fluorescence lifetime in the Fourier domain, defines the so-called metabolic trajectory, okay? So typically when you have your unpolarized cells, macrophages, they can go either way. The whole population lies somewhere in the middle. And if you find your middle there, and then you polarize your macrophages so that they are glycolytic, you see that this shifts to the low end. And if you polarize them so that they become anti-inflammatory, this M2 oxidative phosphorylation phenotype, your fluorescence lifetime goes towards the high end. So you always have a ratio between bound and free, but then it will go one way or another depending on how much of that analyte is in your cell. And so I thought this is all fine and well to detect in cell cultures. It's very useful to do basic cell biology. But can we detect this in thick tissues? Can we detect the same metabolic changes in tissues and actually be able to say anything about disease appearing before it's too late? Before we get there, and that's kind of what the project I'm going to talk about, I want to go a step back and go tell you a little bit about fluorescence and what fluorescence lifetime is. So this is a tissue. This is how complex tissues are, right? We have 4 components. We have some cells, and we have protein. I'm doing the same thing. Sorry. We have the structural proteins. And what we can do is excite with a laser and at least it's fluorescence. So I want to just be very clear here that we have no labels, no dyes, no anything. We just have the tissue itself. We're exciting its natural fluorescence, and we're getting some of this back. What is fluorescing in the tissue? Well, a number of things. The NADH I just told you about, other metabolic cofactors like flavins, but also fatty acids, vitamin A's, which is another kind of fatty lipids, proteins, structural proteins like collagen and elastin fluoresce, lipofuscins, lipopigments, and porphyrins. And what you can see here is that, yeah, you could perhaps identify one thing from another looking at the spectra, but they are, maybe with the exception of porphyrin, pretty boring, very overlapping spectra. So specificity is not great when it just comes to looking at the spectral resolution. However, only based on that, there's already quite some work. Yes?
What's the sort of depth that we're talking about here?
That will depend. This is just really a cartoon, but it will depend on the type of light that you send in. So if you send ultraviolet light, you'll get maybe 100 microns, 200 microns. If you send infrared, you'll go quite deeper, beyond 500 microns, well beyond. If you go 3 photon, you can go even deeper, right? So yeah, it depends. Yeah. So yeah, not ideal. That's why we're interested in adding the fluorescence lifetime. Fluorescence lifetime is really the average time that an electron remains in the excited state. So you have your ground state here, very simplified Jablonski diagram. You send your electrons up in your excited state, and then before they go up in a radiative way, emitting a photon of fluorescence, you have some resident time here. And this is what we call the fluorescence lifetime, and this is what we want to measure. And so you have here schematic, you send your pulse, and after that, you not only have an intensity of this fluorescence, but you can resolve it. It's temporal dynamics, and you get this decay that's typically of an exponential shape.
Or this is something you do during colonoscopy?
Well, if you want to access the gut, then we do it during colonoscopy. Yeah, it doesn't go through all the way from outside. It's not like MRI or PET or anything like that. Yeah, you have to be very close to your tissue. Yeah. So advantages of fluorescence lifetime, it's inherently ratiometric. What does that mean? That depends on the intensity decay over time. So you have a reference point. It's not just an intensity value. It is also relatively insensitive to intensity artifacts, meaning that if you have uneven fluorophore concentrations, you're not going to detect that. It also is more grateful to illumination detection geometry changes, whereas with intensity, if you are not always in the same way, you are already detecting changes on that. That's not really from your sample. And most importantly, it provides an extra dimension to your measurements. It may increase your sensitivity and or your specificity, depending on what you're interested in. Disadvantages is that you have to be a lot more careful now with your experiments. You have to calibrate things a lot better. You need your IRF that needs to be carefully considered into your data. You have to also be careful with the interpretation, because whatever background you may get or count rates need to be within certain parameters, so you're not biasing your results. And analysis becomes a little bit more complex, especially if you have a lot of different fluorophores in your mixture, which is the case that you find in the tissues. Also, it is slow, because you need the time to wait to get all these photons to resolve their lifetime. And always these lifetimes in tissues, we're talking about nanoseconds, anything from hundreds of picoseconds all the way to 5, 6, 7 nanoseconds, depending on what you're looking at. Protoporphyrin, as an example, has a very long lifetime. It's like about 16 nanoseconds when it's in solution. We find that in tissue, in 5-LA, I'm regressing now, but we found about 12 to 14 nanoseconds. That's the longest we've detected so far. Typically, we are in the 3 to 6 nanosecond range. All right, so how do we measure fluorescence lifetime? Oh, okay, that came out weird. We can do two time-correlated single photon counting. That is the gold standard. What's been more widely used. It's a statistical technique in which you are sending photons and you're counting every photon that comes back one after the other until you have enough to put together. You're counting the time your photon comes back until you have enough to put together a histogram from which you can derive your statistics. And out of these curves, you can do fitting and you get your lifetime out of that. There's also frequency domain fluorescence lifetime, which is very common. And in this case, you are modulating the laser that you send in, and you detect the change in magnitude and phase shift in your response. And from there, you derive the fluorescence lifetime properties of your sample. This is what's very typically used with phasor analysis, because you're already kind of working on the Fourier domain. And then there's pulse sampling, and pulse sampling is the technique that I'm going to be using in the rest of the talk for the experiments I'm going to show. And in this case, what we do is we send laser pulses that are of more intense. And what we do is generate the entire decay, which we digitize very quickly at very high speed. So the disadvantage of that is that it requires very expensive high speed electronics. But it works well, especially when you go to clinical applications, when you have all sorts of room light that you cannot control for, which would be very detrimental for approaches like this, where every photon counts. So in the rest of the talk, I'm going to present you with two applications. Both of them are work in progress. And so I'm just going to share what I have about that. And as I said, I'm interested in gut health, as I motivated at the beginning. And the first one is going to be an inflammatory disease model that we're doing in mice. And the second one is going to be the colorectal cancer using colonoscopy and going all the way into human colons to see how disease looks like in there. So the first one, the goal is to develop a system in which we can go inside of the mice colon and we can image as much of it as possible so that we have a tool to follow disease and monitor disease over time, and then have ultimately a tool to do more fundamental studies about what's changing in the tissue. And what we have at the moment is this system that we put together. It has this 355 nanometer laser pulse that we send into the tissue. We send the light out on a side viewing probe, and here I have this schematic of the probe. This is a torque coil spacer and a freeform micro-optic that we attach. So this is the micro-optic that we attach right here. This is the torque coil, a picture of it. And then we rotate this and pull it back. And therefore, we are acquiring images of the entire colon, or as big as we more or less want. Yeah, with a resolution that's mesoscopic. So we're not looking at cellular resolution here, hundreds of microns resolution. And then we collect the fluorescence back through the same fiber, and we send it into, in this case, 2 different detectors. And these detectors are avalanche photodiodes we're using. And then the photons get converted into electrons, and we read that with high-speed digitizers that give us the digitized decay of the fluorescence. And so this was the first version that we had. We were driving the fiber with a motor drive that was borrowed from a cardiovascular application. And if you can see here, the little tiny dot, how we're pulling back here. And this is just real time. And we're rotating and pulling back, and therefore imaging. This was the entire colon, about 70 millimeters in this case. And so what we did next is change this motor drive, because it was a little clunky, with a rotary collimator here, and then we put a linear stage. So now we're doing the rotation through this rotary collimator, and then this translating or pulling the sheath of a catheter with a linear stage. And this is kind of the setup in the lab when we were doing the ex vivo experiments, but the goal was to do in vivo experiments. So then we packaged everything into a card. We polished and finished our catheters, and we encapsulated them with a glass rod so we could have non-traumatic insertion in the animals. And finally, we're doing in vivo animal work over here. And what we're doing right now is we do in vivo, and then we take the colon out and we image ex vivo as well. So I wanted to share some of the results that we're getting with that system. The first thing that I wanted to know is, are we any sensitive to changes? Well, what I want to know is whether with fluorescence lifetime, we can detect the metabolic change, subtle metabolic changes in the tissue, because that's kind of the first thing that happens before any disease manifests grossly. But I wanted to know, is there going to be any change at all, any contrast, right? So we went for a very harsh model, I would say, of inflammation that was induced by dextran sodium sulfate. This DSS kind of disrupts the mucosa completely, and it results in permeabilization. Oh, so the bacteria and all the microorganisms kind of can permeate through the tissue, and it results in very bad chemically induced colitis. But basically, these cartoons here is what you saw before. When they're untreated, these are supposed to be in situ type, doing oxidative phosphorylation. When we treat them with DSS, we create this very harsh, acute state of inflammation. And when we treat them with 5-aminosalicylic acid, this has anti-inflammatory effect. So we are expecting to reestablish the normal epithelial activity and hypoxia. And so this is what we saw. We saw this again is the entire colon. Well, in this case, it was 50 millimeters. Most of them, this was a little longer. But this is the pullback, and then here would be the angle. I'm just flattening out the images. And this is the control animal. This is the DSS group. This is when we treated with the drug, and this was just the drug itself as a control. And we saw that all of the mice kind of behave in more or less magnitude, right? Behave the same way, and the treated mice with the drug were all sort of coming back, recovering from that. And we saw the changes in the, not only the colon length, which is, again, one of the markers for that the disease, or the DSS actually did what it was supposed to do, but we also saw lactate going up, ATP going down, PDH going down, all these markers of glycolytic activity reacting to the disease as we were expecting. However, DSS, as I was saying, it's not just the change in metabolic state. DSS destroys completely the the wall. And there's all these location of the bacteria. There's a lot of bleeding. It's really, really messy. So it's not really the best model if you want to see if there's only a metabolic change. So now we are looking into a model of low-grade inflammation that's induced by overnight treatment with streptomycin. So we are creating a transient dysbiotic state in which we deplete certain bacteria from the colon, and that creates a mild, very low-grade inflammation in the gut, and we want to see if we can detect that. This self-recovers after 5 days with no problem, typically. And so this is what we saw. These are 3 spectral channels that I'm showing here on the device. The device I show you had 2 spectral channels. We just changed one filter to have an extra one. But then basically channel 1, where we expect most of the collagen and most of the structural proteins to show up, doesn't display a big difference. And you can see here the 2 groups. The other 2 channels where we expect metabolic factors to fluoresce more prominently have a decrease in the fluorescence lifetime, which indicates that perhaps we are detecting this increase in free and ADH, for example, rather than the bound one. So there is a clear change in the fluorescence lifetime. We did see that the treatment were efficient as expected, and we saw also some damage in the colonocytes, but not in the rest of the tissue through H&E. And so we did that ex vivo. All of this is ex vivo, and now we want to do it in vivo. And once again, we repeat the experiment, and ex vivo, we saw the expected trend with the streptomycin group going down in lifetime in the stacked channels. And with the in vivo one, it's like, oh, what's happening? So unfortunately, I don't have yet better data for you. This is what I have right now. We're analyzing a lot of data that we took in that one experiment. But basically, we have a lot of challenges here. We have the colons are not as clean as they would be when we take them out and we flash them thoroughly for imaging. There's all sorts of artifacts that we need to understand where they're coming from. And so there is less homogeneity in our colon, which means that we probably get counter, higher regions, lower regions, if we just analyze the same way we're analyzing here. So what we're doing right now is just going and finding the cleaner space and looking at what best we can do about this data. This is where the experiments are at the moment. But in the meantime, another aspect I'm very interested in is what it is really that we are seeing here. And that's a little bit on what we're collaborating with Dr. Sawyer here, and trying to find ways in which we can tell more about what it really is that we are seeing that's causing these changes. And so we're taking these samples, we are bringing them to other imaging modalities. And in this case, we're looking at 2-photon excited fluorescence and SAG, so we can actually see how much collagen there was, whether it was disrupted or not, what's happening with the fluorescence of much narrower bands than what we have in our system, with microscopic resolution, which is what we don't have there. So again, this is work in progress. I just have a couple of images at the moment. And we're also starting to study what can we do or explore what can we do with spatial transcriptomics. So again, we get these rings. This is a cross-section of the colons that I just showed. And we're running this experiment where we're looking at what's happening at the submucosa, what's happening at the colonocytes from the transcriptomic level. And again, an experiment that we are diving into the data on at the moment, but suffice it to say that we have started to do correlations between the FLIm parameters and the genes that we see, or transcripts that we see that are upregulated, downregulated. And we're starting to see some trends. For example, we see this NDOG gene, which is involved in oxidative stress-mediated apoptosis that has it's upregulated on the treatment cases. So we have there a lot more apoptosis reacting due to this inflammation. And we see that the fluorescence lifetime decreases with that. We don't see that with other markers, with other genes. So we are trying to explore whether we can use this as a way to go back and validate what is really correlating with the changes in fluorescence, because it may be what I explained at the beginning on the whole glycolysis versus oxidative phosphorylation, but NADH is involved in so many other things that it could be also something else at the same time. So again, work in progress on that end. And I'm going to be fast rushing. I'm going to move on to the next one, the next application, which is on the colorectal cancer. Any questions before I continue until here? Yeah.
The mouse in vivo study that kind of showed interesting phenomena. Did you have a chance to look at the excising tissue with the same system? And then what was that?
Yeah, this is ex vivo right after I imaged these guys. Yeah.
But then once you take it out,
Yeah.
it changes the lifetime.
So I don't know that it changes the lifetime.
Yeah.
That's for sure, because the lifetime is sensitive to the microenvironment, right? And so you're detecting everything will influence it one way or another. And we know that also from other studies. But there's some trends that should remain not constant, but replicable, if you will. The thing is, though, that here I was doing a very, if you will, lazy analysis, because I'm just taking a square here or everything, and I'm just measuring the mean, right, at the end of the day. I cannot do that here. That's probably a fecal pellet. Then there's this thing here, I don't know what that is, right? There's all sorts of stuff that is distorting my signal. And so I need to investigate that deeper to know, well, first of all, and actually I had a meeting with Jennifer Barton, gave me an idea earlier today that I want to try, but we need to find a different way to analyze. It's not going to work with the same codes that I have for that. And that's why we're kind of in the middle of this. We did this experiment before the conferences. So it's been a while.
Filters that you're using,
Yes.
what's the line width?
Yes. So this is, should have put that in there. This is 390/40, so plus minus 20. This is very narrow, 470/28. And this is 540/50, so plus minus 25. This is kind of broad. It captures a lot of things. This is the narrowest one.
And spectra overlap.
Yeah.
So how do you distinguish them, just by the lifetime?
Yeah, so this is, I'm just showing lifetime here in every band. There is, so I'm not saying, I don't know really what's giving this fluorescence, is the tissue itself. Typically we see a peak in the NADH fluorescence right here, but it's very broad and it does bleed through into here. I also think we have quite a bleed through from the collagen in here that we most likely don't see there. So that's why we need better tools for validating what it is really that we're detecting at every single channel. And that would be the same case, whatever wavelength I choose here, because I'm just exciting with a very unspecific lifetime, wavelength at this point. One thing that I want to do in the future is excite with a second wavelength at 445 or so, where I'll be more targeting flavins in this case. And that's been shown to be a little bit more specific.
Just a quick question on you're showing kind of image analysis where you're essentially averaging the region data.
Yeah.
Have you looked at ratio in these at all? Because you get some of the patterns that appear in both pellet, of course you can't get rid of, but that like band that you see popping up, I wonder if that is that something that could be
maybe
ratioed out?
Maybe. We have to look at all the options right now, yeah.
Quick question.
Yeah.
I'm totally new to this, but you mentioned changing the procedure of removal. Is it when you're doing it in vivo and then ex vivo, is it getting squeezed when you pull it out?
The pollen?
Yeah. Is that why this pattern may have changed? Or
So what changes is that we flush quite a bit when we take it out. And so we really clean whatever is left behind.
I see.
Yeah. And we probably damage the mucosa while we're cleaning. Honestly, I'm not sure. We want to run a couple of experiments to see how much damage we're making while we do all these steps. So one of the things that we try doing is clearing the colons ahead of time with some sort of sugars to kind of have cleaner space, right? And then I saw fluorescence, a different fluorescence response in the tissue. And that kind of defeats the purpose. So tricks that would be useful for other, like OCT, for example, or other things, other modalities that would be useful there, it's kind of playing, it's not helping in this case. So we have to find also maybe an agent that works better for us. We also don't want to put them in a too harsh of a fasting diet because that may affect the metabolism itself. So if we want to study metabolism without altering it, it's tricky. Yeah, tricky. All right, so let me just tell you a little bit about the colorectal cancer study. And this is just a pilot study that we done this year. But yeah, I think we were talking about this earlier. Colorectal cancer is the third more diagnosed cancer worldwide and the second cause of cancer-related death. And most problematically, there is an increase in incidence of early detection of early onset of colorectal cancer. So something is happening apparently. There's also still very low rate of early detection. So there's a lot of room for improvement in there. And so what happens now, well, it's screening. Screening can prevent colorectal cancer. This actually is something that you see on the cancer.gov. It's being credited for lowering the rates of colorectal cancer. It's so important, right? And when we go there, the goal is to detect disease that's small, that hasn't spread much, and that's supposedly easier to treat. And one of the things that they do right now with screening, they go in and whatever polyp they find, I don't know if I can play this again, but you saw the little arm going out and they take the polyp and they take it and it's out of the way, right? And then they biopsy it and they see whether it was malignant, it was benign, or what kind of polyp it was. But you know, a polyp can take many years actually to develop into cancer, if it does at all. And in situ polyp identification would allow to remove just the high-risk tissue, just the tissue that is malignant and that poses a problem, perhaps then reducing the patient discomfort, any complications that can come out of that, any possible morbidities that come with it. And will also alleviate the burden on the healthcare system. I mean, right now, all of these single biopsies are costing it's a lot of work to process, and it's also very expensive. So having a way to identify whether, you find your polyp here, and it tells you already, oh, this is a hyperplastic polyp, or this is a tubular adenoma. Careful with that, right? It will be very cool. It will be very useful for clinicians to support decision-making, to detect early cancer, and to assist also in surveillance protocols. You may be willing to say, okay, let's just keep watching this guy over a course of time, whatever is sensitive, and see how it evolves, right? So that would be nice. So that's kind of the vision, right, where we're trying to go. And we put together this system, which is a version of what I showed you before. We connected it and, with commercial colonoscopes. Basically, we are putting there a fiber that we're threading through a commercial colonoscope, and we're going into patients. So this system, all the optics are happening in this box over here. What you see here is the very expensive digitizer to get the readings of the decay. And everything that's in this box is really, this is a render of it, but this is the schematic. You have the 355 nanometer pulse lasers that go into your fiber, the fiber that goes into the colonoscope. You get the fluorescence back, and we separated in this case into 3 different channels at once. Again, the channels being the 390/40, the 470/28, and the 5, in this case, 42/50. We have the 3 APDs and the digitizer. We also have here a aiming laser. We interleave that in so that, and you'll see in the pictures and maybe even here, this blue dot or white dot, this is the aiming beam laser that we could say. And we need this to know where it is that we acquire data from. Otherwise, we wouldn't be able to see where we are acquiring our data. And it will be very hard to know whether we are in the polyp or not, because this moves a lot. And it's very difficult to control. And typically what you see is like, we're out winning. So it's a little bit of a challenge there. So what we're doing is acquiring this data, then they resect the tissue as per normal protocol, they go to histopathology and we get that result. And so we are compiling a data set of fluorescence lifetime parameters, metrics, and whatever polyp types the doctors tell us it was. And we are sampling this library. We have a number of polyps and normals that we've acquired from all over the colon in different areas. And I just want to show you a couple of examples of what we're seeing today. One example, and you see here, one of the examples with the motion. This is normally looking adjacent mucosa, and you see how it moves, but it's fine because it's all normal mucosa, right? And here there's a very large polyp. I'm hoping you're able to see over here. And again, it moves a lot. In this case, it's so big that this really doesn't matter. We are on top of it all the time. And so then we're extracting these parameters, we're putting the lifetime together, and the first thing we see is that these are huge error bars to begin with. But we do see a trend between longer lifetimes in the adjacent mucosa and the reduced lifetime that we're kind of expecting on the polyp. This polyp turned out to be a high-grade dysplasia polyp, tubulovidous adenoma with high-grade dysplasia, which is malignant, and this patient was there for a resection, so this polyp went out of the way. We also observe a little bit of a red shift, actually, even it's looks mild here, but if you look at the standard deviation, it's more significant than what we see here, where the fluorescent, and this is intensity ratios of what I got in the first channel, going down in the polyp and going up in the far wavelengths, meaning that there's a red shift overall in the spectrum, which is also, we keep seeing that in all the other cancer projects, seems to be a marker or something indicator of disease, cancer disease in this case. So we have these kind of, we actually have just one of these big polyps, and then we have a bunch of small polyps that we find on routine colonoscopy procedures. And these are just two examples. In this case, we went in and we found a polyp in the cecum that turned out to be a colitis injury lesion. We then found a polyp in the descending colon that was a tubular adenoma, and another polyp in the sigmoid that was a hyperplastic polyp. So 3 different types of polyps. And if we look at the data, now we're just looking at 2 of the metabolic channels, we see that the mucosa was kind of pretty consistent, and consistently we see a decrease in the polyp signal in the lifetimes, but between them, they don't look really very different. So good, we can see that there is a polyp, but we can also see that visually. And there doesn't seem to be a lot of difference between them. Whereas here, we have 3 polyps again, 2 of them being tubular adenomas and another being a hyperplastic polyp. The ones that look more different are actually the tubular adenomas. So it's like, okay, interesting. Again, we're just collecting data there. So, but I still want to know whether we can use this kind of data to ID what kind of polyp we have. So I'm looking at all this data together. For the type of polyps that we're encountering the most are hyperplastic polyps, which are the least malignant. I would say they are not even considered malignant at this point. The tubular adenomas, which by being an adenoma, they are already careful, it's precancerous. And then cis-ulcerated adenomas. And this is and green is the normal mucosa. If you look at this like that, it doesn't really say much. It's not very encouraging. So what I did is look at just the normals, and the normal mucosa across all the patients that I have across the entire colon length, and we have quite a big spread of values. And I'm like, what is this? Why so different, right? It doesn't seem to be actually a difference that's along the colon, but every area is very spread out. So I'm looking at the patient themselves, and I see that some patients have a very long baseline, where others are very low, and anything in between. It's a very big spread between 3 to 6 nanoseconds in this channel, a little tighter in the other channels, but still very big variations. Within one patient, then the standard deviations are a lot smaller, but there is very high interpatient variability. So what I'm decided to do right now is we are taking the data from the polyp, and we're always imaging the adjacent normal mucosa. So we are doing a relative lifetime measurement in this case. And now we start to see some interesting trends. We actually see that in the 390 channel, the hyperplastic polyps, I have 7 of them. They are always positive, where the other kinds are mostly negative, some of them going up as well. The hyperplastic polyps are going mainly negative on the other two channels. And there's a slightly opposing trend with the Sissels-rated adenomas. Again, numbers are underwhelming, but we're collecting data as we speak. So hopefully we'll be able to enlarge the library and say a little bit more about what's happening there. We have to find more clever ways to analyze this data to kind of get rid of all this interpatient variability. Also, there may be intra-patient variability that we see across the colon of the same patient. So we'll see. And so the ultimate goal is actually to be able to detect cancer. So we need to know what cancer looks like as well. And I just have one patient here, and this is just almost raw data that I'm displaying because I only have this one case so far. Here's the tumor. We did find a small polyp in that patient as well. So I'm displaying that. You see that the cancer or the tumor in this case has a different signature than the rest. So it gives us some hope that there should be some sort of, there's an angle that looks different than the normal in the beginning. And this was a hyperplastic polyp. So a very different kind of lesion. And so ultimately what we want, or what we're trying to do, is to study all sorts of different gastrointestinal health problems, or we're trying to find a method that can tell us, that we can use to study gastrointestinal health. And go ahead.
Also tumors, but earlier you said that tumors form in polyps first. So how far into the progression from polyp to full-on tumor are these tumors? Do you see, like, is 50% of the cells in, say, a specific clump still a polyp as it's turning into a tumor? Is there a way to sort of like differentiate that clinically?
Yeah, I don't know until you open it up and you see it histologically. You cannot tell whether there were cancer cells in there or not. I'm not quite sure, though I understand your question. You want to know?
Yeah.
Mm-hmm.
So, like, obviously the bottom one, they, excised it and then, did histological measurements and found it's tumorous.
Yeah.
I guess the question is, like, from it being a polyp to
from
becoming
here
the
to here?
tumor,
Yeah.
like, what stage of tumor development is it actually at that point? Is it still 50% polyp tissue, or is
Well,
it?
yeah. So this is their scales, and this is well characterized. Well, as well as it can be, right? At this point, I guess the problem being you don't go there like every day or every year even to test what's happening or how it has evolved and how it has morphed. So I guess by histological features, they've put out these scales and these ranges of tumor. And to be honest, I don't know in this particular case what was the stage of this tumor. It could be a very advanced one, or maybe not. I don't have that information yet. I don't have the pathology for the tumor part of here yet. So yeah, I'm not sure. So yeah, tools that can help us understand this. And what I think fluorescence lifetime can do, if we can demonstrate that like they do in the cells and the organoids, we have an opportunity to be able to detect metabolic changes in tissue. We just have to really carefully characterize it and see what would be the parameters and what would be the configuration that will give us that information. And so what we're trying to do is a bunch of mouse experiments so that we can test different scenarios. In the meantime, we're gathering patient data because otherwise we don't know what to expect when we go there, which we know will be very different from what we see here. And so eventually we can look at things like inflammatory disease and colorectal cancer. Inflammatory disease really preceding colorectal cancer, but we can also look at infectious diseases and overall actually have a tool to study microbiome changes. So with that, I have a wish list. I thought that this would be, a good audience for that, which is I'd like to increase the spatial resolution of intraluminal FLIm imaging. I think multiscale endoscopy could be a way, for example, with the mother-baby endoscopes, for example, or tip on the tip kind of configurations, maybe, where we can do fast mapping for the low resolution fluorescence lifetime, but large field of view, so we can find like hotspot areas or problematic areas, and then go microscopic resolution wise, high resolution, this should be an S, high resolution, but smaller field of view in different places. I mean, if we have a way to image the whole thing with microscopic resolution, would be my guess. But that would be like a tool for longitudinal mechanistic studies. The idea is that we don't have to sacrifice every single animal that's giving you like beautiful, nice data just to start all over again with the next one. So we need some tools to follow this development without having to chop up the tissue every time. I would also love to have wide field FLIm colonoscopy. So you've seen these images, and I think there is an opportunity. If I go back even just here, this thing moves a lot, and you saw a little bit of it here, just an example, right? And these are pretty mild cases. But at some point, there is some stillness, and it could be an opportunity to just take a snapshot and do some sort of wider field, maybe with sputter arrays or some other cool detector systems that are going on right now in the field. Yeah, so for more higher throughput and also friendlier use. We're now putting the fiber in, fiber out, and it's kind of a little cumbersome. And yeah, finally, the multimodal, getting intrinsic multimodal content. With this, perhaps we can say things about energy metabolism and perhaps other aspects as well. But if we add Raman to it, we can say things about what's happening with lipid metabolism. If we can add SAG, we can say things about what's going on with the tissue structure. And all of these will provide a lot more information for early detector markers that can come from either of them or a combination of them. And so that would be very good for early detection, also for monitoring therapies, see how things are going over time, kind of quality control, assurance, and whatnot. Yeah, and with that, I just want to thank everyone who's helping with this project, everyone at the biomedical engineering babies, as well as in the health system, the funding agencies that are making this possible, and all of you for your attention. Thank you. Thank you very much.
All right. Thank you very much, Alba. Questions from the audience. DK, do you want to start us off?
Yes, very nice book. So two questions. What is the power, illumination power of the tissue with 355 nanometer?
Yeah, with the mice, we have at the tip of the fiber 0.5 milliwatt. Yeah.
Similar level for the human patient.
Yeah, it's not very different. Yeah.
The other question is that so for your lifetime estimation, I guess it was assumed that there's only one, lifetime. So basically, single,
Yeah, no.
lifetime. So
Yeah, that's a very good question. Thank you. So we are not fitting a mono exponential decay. We're also not fitting a bio-exponential decay. We're not fitting exponential decays. What we're doing with this data that I showed, the way I'm extracting the lifetime, and the conversation we just had with Thomas and Dr. Schreier earlier, we are doing what's called the Laguerre, least squares Laguerre expansion. Okay, let me just tell you how it works, and you'll understand very quickly. So we get this decay that obviously has the influence of the IRF. So what we're doing is we convolve the IRF with a basis set of Laguerre polynomials that they have a decaying profile. And you can choose how many you put. I'll give you different accuracies, I guess. But then basically we convolve that, and then we deconvolve our signal with that base, or we expand, sorry, our signal into that basis set of polynomials. And from there, we get all these coefficients, and we can fit basically the decay. We can approximate the decay with a series of polynomials. And out of that, we get the lifetime so from now we get the decay, and out of the, let's say, I want to write, out of the reconstructed decay, we get the intensity under the curve, or the intensity, and then we get the probability density, which would be the lifetime of the decay.
Well, I was wondering, so things like the phasor.
Yeah, so exactly, we can do all of it. We could do multi-exponential fitting as well. We haven't yet tried that. but we are actually just trying the Fourier transformation. Once we have the decay reconstructed, then we can do whatever you want with that. And so we could fit that, or we could Fourier transform and get the phasers.
So because I mean I guess the area of the volume of the tissue that's generating fluorescence signal for you is relatively large, and it might not be all the homogeneous, right? So you might have like a mixture of the different lifetimes coming along. So then, like, if there's a tool, for kind of looking at the spread of it, I mean, which might be essentially the probability function for the lifetime, right? So, but then what we were kind of seeing is for each image, you're eventually presenting the average or like a peak in the lifetime probability function, or I see.
So what I was showing here was just one value. Why? Average of the whole thing, because from the biochemical analysis, I get one value, the lactate level of that colon. And so what we needed to do is to match the two. But what I want to do with spectral transcriptomics and the other imaging modalities is, can we break it down and go to a point where we can look, maybe not pixel to pixel at this point because we don't have matching resolution, but a little bit of a spatial discrimination. Going back to the phasers, I've looked at the data in the phasers, and at the end of the day, at this point, I do see that if I see a change with these metrics that I extract from the Lagrange convolution, I see the same in the phasers. It just gives me a different visualization, but it doesn't give me anything new, because I think we haven't worked out a way to calibrate, and what would be interesting to see, we haven't defined the metabolic trajectory of what we're finding in there. So I need to measure that and set up the entire experiment for it to make sense, for us to maybe learn anything from that type of analysis. But yeah, you're right that we could be studying the spread more carefully. We should.
It's established that the fluorescence or lifetime studies are the best way to do diagnostics on these polyps. For example, how about the mechanical properties of the polyps? You press them mechanically, and you look at the elasticity, for example. Could that indicate that they are precancerous or not?
Maybe, I'm not aware of that.
Or if, usually cancerous cells attract a lot of blood, so if you could measure the blood flow. Optically or otherwise. Is that established that fluorescence lifetime is the best way?
I don't think it's established anything right now is the best way to look at this. Right now, the gold standard is white light endoscopy and narrowband imaging, which helps because of the blood. Sometimes if it's very clear differentiation, then the narrowband imaging helps quite a bit if there's a lot more vasculature presence in that area. But that does not help across the board. So right now there is not, let's say, one technique that's established to be the best to detect the polyps. Every polyp they see right now, they just take. There's quite a bit of flat polyps that are more difficult to just identify because it's not a mass that's sticking out. And then there's also, kind of going back to the fluorescence, evidence from 20 years ago even, that there is both in the spectra and the lifetime distinct properties with point measurements that were done both in vivo and ex vivo in different models that it indicates that there's difference between the different types. But it's all, again, point measurements. And what I'm doing here is still point measurement, but we're doing it so fast that we have this pseudo capability of imaging. And so that makes it a little easier to acquire data and to run this kind of experiments than with the technology that was available 20 years ago. Yeah, so there is a prospect, but if it's the best or not.
Yeah, and if I could chime in, that's an interesting thing mentioning the elasticity or stiffness. Some of my work does look into that for other cancers as well. A lot of it ends up being the geometry, that if you're doing something where you're looking through the side, it's a lot harder to press on the tissue with a known force as opposed to like looking at it straight on. And so I think that this has the advantage of being kind of the cylindrical coordinate system, and the colon is very much a cylindrical type of object. Kevin.
Can you show the probe system again? You showed like the imaging layout. I just was curious. I understand what you guys are saying. You got to keep it compact. Just had a quick switch.
See, like this one or the other?
Oh, yeah. Actually, so when you add the endoscope into the system and then you add the laser illuminating, is it also the imaging system all in one? Is that an all-in-one system or is it multiple? Because you had a camera and then the probe. So is the probe itself the imaging system or is it?
Yes,
Oh,
the
I see.
probe itself is the imaging. So the camera, I'm using the camera from the colonoscope.
Yes.
So yeah, I'm using the camera from the colonoscope, and then this is nearby the patient, and we have a fiber that's 5 meters long. Just it goes into the working channel of the colonoscope, and then you just see it here. This is the tip of my fiber. This is the tip of my fiber.
What?
The signal is measured whatever fluorescence comes back this way. Yeah. So there's the aiming beam, that tells me you just acquired measurements there. But then whatever fluorescence is so weak that we don't see it, but then it's isotropic. So some of it is gonna go back into the fiber, and that's what we're detecting.
Then was on the receiving end. Have you guys thought about adding grading so you could multiplex the signal and pick up with multiple cameras as opposed to one?
Yeah, that's something we are thinking about. So right now we have 4 channels, actually the latest configuration, we have 4 channels. I've been wanting to put a grading and a spectrometer to look at the entire spectra for a while. It just hasn't happened yet. This is kind of a locked system. Once it's there in the clinics, we should not touch, or we cannot touch its design too much because it's part of long-running studies. So I'm basically borrowing, there's a head and neck study that we're doing, and it's counting more than 200 patients now. And I'm just borrowing the machine. Whatever I have, it's what I have. But for the future designs, yeah, I want to put a spectrometer. And to do spectrally resolved lifetime is a little bit more challenging, and it's perhaps still too slow, except with the spatter rays that I'm trying to learn about. That could be an option. Yeah, thank you.
I had a question on this now that this is up. So you mentioned potentially coming back to monitor the development of polyps or something in patients. I'm curious, how would you go about tagging them so you know which one you looked at last time?
There's some of the things they do right now, they use tattoo ink to mark some things sometimes. That does alter the lifetime, I've seen. But another option is to hope that there's no other that pops up in that area, but that's not a very good scientific option. I don't know. Well, we could put some sort of fiducial, perhaps, if there was something that was not intrusive, I guess the ink is the least invasive one, just maybe somewhere nearby. I don't know. Yeah.
Generate a sort of fiducial that.
I mean, technically you can, but I don't know that you want to do that on the patients necessarily. I think that the clinician I work with would be, but I don't know if it were beneficial. It's a technical option.
No, it does just seem tricky because the colon is such a weird organ.
Yeah.
Interesting.
But yeah, I mean, tattooing ink, it's something that they use routinely. Because, for example, they find one of the big polyps, they mark it wherever, and then the patient comes back again, and then they know where they need to cut, where the tattoo was put.
I see.
Yeah.
Interesting. Right, because potentially the surgery isn't in the same procedure as the colonoscopy. Yeah. That makes sense.
Have you seen any heterogeneity in individual polyps? Is there any concern of missing underlying biology?
Yeah, I haven't yet, but this is something I'm very curious about. How the signal changes around the polyp and even the normal tissue around the polyp. So there's something called the field of cancerization around tumors, right? The tissue reacting to something happening there. I don't know how that is for the polyps. And that's why I would really love to have like a wider field image where I can capture the polyp and the rest and see where the signal starts changing and where don't, and also how homogeneous or heterogeneous the normal mucosa really is. With the in vivo experiments, you cannot really control very much where you're imaging, where you're acquiring data for. That's why I'm trying to work with the mice people to try and develop models in which we can test this hypothesis. And I know that right now it can sound like very disjoint projects, but you start where we can, and then we'll work our way to inform one and the other.
No, well, I think it makes sense because I was also going to ask about, you see a lot of patient-to-patient variability, which I think is pretty common with humans. But kind of connecting the mouse results to the humans, I mean, you could look at whether the humans are on high-fat diets or if they smoke or drink or something like that.
Yeah.
Imagine that might be potentially an explained source of variance or something like that.
Yeah, maybe, and we see that in the other cases as well. And the head and neck, we're seeing that the smokers have a different lifetime than the non-smokers, which I think
The lifetime of the smoker.
much longer. But I will have to check. What I wanted to say is that patient 23 that was so high. It's in the middle of my existence to this day, because I'm like, what is special about that patient? Apparently nothing. I remember one patient that had very pigmented colon. I'm like, that must be it, but it was not that one. The very pigmented colon blended very nicely with the others, and I'm like, oh, shocker. So I really don't know. It's more of a detective job at this point.
Can you not do that also with the grading? Illuminate it with a coherent laser and then just shift the cameras off axis to have synthetically wired field view?
I don't know. Can I? Maybe.
Is that not done? Or
I don't know. I don't know much about it yet, honestly. Maybe. And if it is, I'll be happy to learn about it.
okay, cool.
All right. Any last questions from the audience? All right. Well, thank you again, Alba.